This is an exam application tutorial on the topic of protein synthesis. The question is, explain how the information in a gene is used to make a protein. Quite simply, this could be translated as explain the process of protein synthesis, which is effectively what this is asking. That can be subdivided into two questions, each worth about 3 marks. Explain the process of transcription, the first stage of protein synthesis, and explain the process of translation, the last stage of protein synthesis. So if you remember, the first thing that happens is DNA unzips to expose a gene, and you can see that happening here. So you can see the weak hydrogen bonds break, and now the strands separate out. Now, the information on the gene is copied onto a molecule of mRNA. So these are mRNA nucleotides. You can get them from your food. When you munch food and munch cells in food, you'll basically break up the nucleus, break up the nucleic acids to get a supply of nucleotides. Just like you digest starch to get a supply of glucose. Nucleic acids break down into single units called nucleotides. So mRNA nucleotides will be attracted to their complementary base on DNA in the exposed strand. And you can see that happening now. So you can see nucleotides for mRNA are basically attaching to their, or bonding with their complementary base partner on DNA. But you can say all that just by saying the information on a gene is copied onto a molecule of mRNA, and that's our molecule of mRNA. The final part of transcription is simple. The mRNA leaves the nucleus. So now we can see our mRNA detach and leave the nucleus where it heads into the cytoplasm. So remember, transcription occurs in the nucleus. And now we need to describe translation, which occurs in the cytoplasm. So our mRNA bonds to a ribosome. A ribosome is a part of a cell. There are many ribosomes in a cell, and they all do the same thing. They produce proteins. So the ribosome's job is to actually manufacture proteins. Never say DNA makes proteins. DNA just stores the information to make proteins. So mRNA bonds to the ribosome in the cytoplasm. 1 mark. Now for the most complex sentence in this answer. tRNA anticodons carrying specific amino acids are attracted to their complementary codon on mRNA. So you can see here that I've divided the mRNA into 3 codons, 3 triplets, 1, 2, 3. Now if you look at tRNA, that kind of clover leaf shaped molecule, you can see that it also has 3 bases, a triplet, but we call this triplet the anticodon, and it's complementary to the codon. A pairs with U, C with G, G with C, and so on. Each tRNA molecule will be associated with a specific amino acid. Hence, tRNA anticodons carrying specific amino acids are attracted to their complementary codon on mRNA. If you get this sentence right, everything else should be very easy. So let's play that forward. We can see more tRNA molecules carrying their specific amino acids bonding temporarily with that mRNA molecule. Finally, because we haven't actually made the protein yet, to make the protein, those amino acids must bond together. That's what those purple dots here represent. So the amino acids bond together to make a polypeptide or protein. You can say either. And you can see that the tRNA molecules leave, like taxis, leaving their amino acid passengers behind, bonded together. And that's the word perfect answer for explaining protein synthesis.